There's another polymer that we call proteins. This is the structure of an amino acid. This is an amino acid. When you go to GNC and you read this thing, amino acids, right? These are the, remember in DNA, what's the monomer for DNA? Monomer for DNA. Let's make a chart, because obviously we need to make a chart. What's a monomer for DNA? So let's put all the monomers here. And let's put the polymer here. If the polymer is DNA, what's the monomer? Yeah. Nucleotide. And it's a deoxyribose. Nucleotide. By the way, when somebody, I've been reading your reflections from your quiz applications for getting your 20% back. And people say, I studied. You see, when you study, that word study really doesn't have any meaning to me. And I don't think it has a lot of meaning to you either. I think for a lot of you, studying means you read it. I read it, I looked at it, I paid attention in class, I studied. That's it. That's not enough here. That won't work. You have to take what you learned and you have to organize it. One graphic organizer is a comparison chart like the one we're putting here. Monomer of DNA is a deoxyribose nucleotide. What's a monomer of RNA? RNA is a polymer. What do you think it is? Close. It's a ribo nucleotide. Same thing, right? Except that it's ribose sugar and has a U instead of a T, right? But basically RNA is just a polymer of different set of monomers. There, in both of these cases, we know what the structure is, right? We can even put a structure column. Let's make a structure column. What do we know? Just a basic outline of the structure. What do we know? A pentose sugar, I heard that. Yeah. Perfect. Just draw a pentose, a pentagon, that's fine. Nitrogenous base, perfect. I love it. Let's put some kind of weird ring structure. Let's put a double ring. Nitrogenous base. And a phosphate. Perfect. Perfect. The three parts, phosphate, ribose or oxyribose, right? Let's just say sugar, and nitrogenous base. The three parts of our monomers. Now, depending on which sugar you either get monomer 1 or monomer 2, or right? Either deoxy or ribose. Pretty straightforward. I could add another column, how did we make it? But that's not really that important right now. Everybody got that so far? All right. So now we're going to look at a polymer called polypeptide. It's really messy, I know. It's one of the things kids, unfortunately, I'm trying to write quickly. Yeah, not so accurately called a protein. So proteins are the things you eat. They're made of your muscle. They make your skin up. Your hair is made of protein. Right, there's all kinds of proteins that we call. By the way, this is obviously these two have different functions, and we could add functions to our great list for studying. You know why this is so useful when it comes to studying? Anybody have an idea why it's useful when it comes to studying? Because I can put little cards over it like this, right? I can test myself. So when people tell me on these individual learning objectives that they plan on studying, right, and that they plan on doing better on the next quiz, or when I ask them what they're studying, how do they study? They say, I just read it. They tell me, I ask you, do you understand this? And 90% of the kids will do this, right? They're just not, they won't even say yes. Some will say yes. But if I ask them, how do you know that you understand it? I never get anybody saying, well, I covered up the answers and I was able to tell myself what the correct answer was. Can you do that? Can you cover up an answer and be able to fill it in, say it out loud to yourself or to your friends? Do you sit at lunch in study groups or with a friend and quiz each other? Or do you sit at lunch and talk about nonsense? So, and don't do this. Don't try to quiz yourself right before the test and think it's going to be good enough. You know, what's funny is if you did just a few minutes a day, just 10 minutes a day doing something like this, you'd have it straight. The test would be easy. Instead of spending all night struggling and sweating and then not doing well anyways. Right? You got to become more efficient. That's what we say. That's why we say something like you need to study smart. It's not always about working harder. It's not always about working harder. All right, so let's take a look at a polypeptide. Let's look at a monomer of polypeptide. We'll come back to this chart. We'll fill it in a minute. Let's take a look at it. Look at this monomer. What does anybody see? We talked about two of these functional groups already, didn't we? I said you didn't have to memorize them. You don't. But if you would have memorized I said this one, carboxyl group, is going to be very important. You'll see it a lot. And I said the amine group is another one that you'll see a lot as well. So if you can tell C double bond O, single bond OH, and know that it's a carboxyl group, forms a carboxylic acid, carboxyl. And if you can tell an amine group, it's an NH2, stick in there. Very important little molecules. They become important. This little group, and this nitrogen, NH3, NH4, this is what you produce every time you do, every cell of your body is producing this stuff, and you have to kick it out. You produce ammonia. Ammonia is dangerous, so you change it to urea. It's a salt. Urea you do what with? You pee it out. So this little molecule is very important in the sense of metabolism, in the sense of fertilizer for our fields, in the sense of pollution for our water, in the sense of the stuff that makes up who you are. Very essential. Clearly, carbon, it's part of the schnapps, isn't it? All right, so ammonia, the amine group is not ammonia, it's amine, because it's NH2, not NH4. All right, so it's NH2 but you see they're just related, right? All right, okay, just as long as you're clear on that. Here is carbon in the center. So see how small it is? It's pretty small. But there's a weird little thing that says R. This is not an element. This is a variable. A variable. You know, like X and Y are variables in math. R is a variable here. R groups in biochemistry, organic chemistry, R groups are generally known as side chains or variables. They can be different things. But what stays constant in an amino acid is these three parts: the amine group, the carboxyl group, and the hydrogen. These three stay the same, all around one center carbon. You're going to take and connect a bunch of these together. So we can go back to our chart real quick, and we know that the monomer for polypeptides is what? What's it called? Protein is the polymer. Another word is polypeptide. What is the monomer? Amino acid. There are 21 different amino acids. Remember we talked about hair. We said if you have a lot of cysteine in your hair, what is your hair? Those of you that paid attention to the case study, you actually did it and didn't copy off someone else. Or read the book, read this study guide. What is cysteine? Ouch. That hurts me to the core. Cysteine is sulfur. It has sulfur in it. It makes disulfide bonds. The more cysteine you have in your hair, the curlier your hair is. Do you remember that case study? Disulfide bonds. Cysteine has sulfur in it. All right. Well, cysteine would have one amino acid, an R group that has sulfur in it. If the R group has sulfur in it, and you have a lot of this amino acid in your hair, you can form disulfide bonds and it curls up. If you don't, then your hair is straight. All right. Just trying to make some connections here. By the way, that's the other way you can learn better and be smarter and be able to be more adaptive. And that's by making connections in the different things that you learn. You spent an entire day while I was gone doing a case study on hair. Did you think I was wasting your time? I heard yes. I'm sorry that you thought that. I never waste your time. Just like I'd never want you to waste mine. Go back and look at that case study, which, by the way, some of you still have to upload. They're zeros until you do. So you have a carbon in the center. I'm not going to spend the day discussing that. You have a carbon in the center for this amino acid. On this side, you have a carboxyl group, which looks like this. Up here, you have a variable that we call an R group. Down here, you have a hydrogen. And over here, you have an amine group, NH2. That's an amino acid. This R group will be different depending on what amino acid you have. If you have cysteine, this R group will have what? Sulfur. Excellent. And if you have a lot of this cysteine, then you'll have curly hair. Naturally curly hair. If you don't have a lot of it, then you'll have straighter hair. So that's, well, I'm about to show it to you over here. So this is what it looks like again. I just drew it, but there it is, the carboxyl group, the hydrogen, the R group, the amine group. It looks like this in a space filling model. Do you see why we draw it this way? I don't know about you, but I like this better. I can think about it better. I can identify different components better. This is really important, though. Space filling models are very important when you're trying to place things together. You have a computer program that wants to see how they're going to look, how the protein's going to look, and predict how the protein's going to look once you put them all together. It's really nice to have a space filling model to do that. And I'll show you some pictures of some of those space filling models of various proteins that we've created using these computer algorithms, as well as X-ray crystallography. So that's structure of amino acids and general structure of amino acids such. Are we good? general structure. Let's look at a specific. So each of the properties, there's, what is this? What's this? How do you pronounce that? Cysteine. What does it have? Huh? Sulfur. Oh, that was sarcasm in case you missed it. No. It can do what? What can it do? What can it do? Oh yeah, if I only would have read the study guide, man. I'd be ready. Disulfide bridges. Disulfide bridges. This is what makes your hair curly. They cross-link. It's what bends these things. Otherwise, you'd have giant straight proteins all the time. What's bending it? What makes something bend versus not bend? It's things like this. Why? Because they interact with one another. We're going to look at it in just a minute. Here's another R group. This is one. Look at this one. This is longer. That's going to influence the shape, isn't it? If something's tall and long. I saw a kid get out of a car the other day, just yesterday. We were at Mark's, my wife and I, and the kid got out of the car. I kid you not, this kid was 8 foot tall. I thought to myself, man, him in the car and me in the car, we just would be whole different configurations. You know what I'm saying? I'm round, he's tall. It's just weird. The really long chain, there are longer ones, but this is a lot longer than this one. This is small, but this is really important too, doesn't it? Each one has a different property. This one makes disulfide bridges. It'll kink the protein or kink it, meaning bend it. And this one makes it alkaline, which means it's a base. This has ammonium at the top, or an amine group at the top here, so it actually will act as a base. It'll be, that pH, the pH scale we talked about, where you have low pH and high pH. This makes the protein have a higher pH. Yep. Alkaline and water. Where do you see that?
No, I was asking a question about does it have that formula?
Does alkaline water? Water is neutral by definition, so it's both base and acid. So it's neither. So they equal each other out. The acid form of water and the basic form of water equal each other. They're in equal amounts. Here, this substance is alkaline. Did I answer your question? I'm not sure I understood it, but okay. So this is alkaline. So this is base, and this causes kinks in the protein. This causes it to have basic properties, right? Act like a base. This is a carboxyl group or carboxylic acid group, and it's going to make what? The protein a little more acidic. So this makes it a little basic, this makes it a little acidic. If you have a lot of these, the protein's acidic. If you have a lot of these, you have the protein's basic. You have a lot of these, you have a really curled up protein. You don't have a lot of these, you have a straighter protein. Yes, question? Yes. So the properties of amino acids come from their R chains, because each of the amino acids are all the same for everything except what? They're R groups. So these R groups make them different. They change how that amino acid's gonna, what qualities it's gonna give to a protein. So if you have a hemoglobin, which is a protein that's in your blood, right, that carries oxygen, it's in your red blood cells. If you have 4 hemoglobins and you get the proper shape and the proper qualities to each amino acid, the oxygen's carried no problem from your lungs to your tissues, released, everybody's happy. That cycle goes on for 3 months, your red blood cell dies, a new one's born, you use the iron in your blood that you eat to produce more hemoglobin, and you keep going until the day you die. Life is good. If there's something wrong, and just one, by the way, this is what happens in this disease. Just one single amino acid is changed. Instead of having this amino acid, you have that. You're gonna know which one, because we're gonna actually look at it, we're gonna look at it deep. You just change one amino acid. Hemoglobin doesn't work anymore as well as it used to. We call it sickle cell. And hemoglobin doesn't grab onto the oxygen well, which makes the cell sickle, which makes the person anemic. That's why it's called sickle cell anemia. So one change in an amino acid could lead to big changes in your life. One mutation, one amino acid change, now you have sickle cell trait. If you have 2 copies of that gene, you have sickle cell disease. You have pain and suffering, and you have to take medication. God willing, you'll live a normal-ish life. I'm sure there's a lot of success stories of people with sickle cell disease, but it's hard. It's a hard disease. Sickle cell trait, you don't have a lot of symptoms, but having sickle cell disease can lead to a hard life. My best friend in high school died with sickle cell disease. He died right after he had his son. He was 19 years old. So it's not, and we're coming up with ways of curing this. We have things called CRISPR that can make changes to the DNA. And by the way, there's another question. Let's go on, and we'll talk more about that as we go on. So when we talk about these connections, these are all connected. These are monomers, right? And we're going to do what with these monomers? What do we do with monomers? Connect them together to make what? Polymers. Monomers, polymers, monomers, polymers, monomers, polymers. These are the Legos. We put them together to make our thing. With DNA, what was our monomer? What was our monomer? The nucleotide, either deoxyribonucleotide, correct. If you had RNA, you had ribonucleotide. If you had a polypeptide, or what we call a protein can be more than one polypeptide. That's why I say it's kind of a protein. If you have a polypeptide, you have what is the monomer? Amino acids. So with every monomer, you put a bunch of the monomers together, you get the polymer. That's the relationship between the two. That's it. Take a bunch of these, put them together, you get that. How do we do it? What is the process? What do we call it? By the way, you're going to get these questions on a quiz, so make sure you're writing them down and you know how to answer them. Put them on index cards, tattoo them on your forehead, whatever works for you. If you tattoo them on your forehead, make sure it's backwards so that when you look in the mirror, you can read them straight, right? Don't actually do that. I hope no one goes home and tries to tattoo that answer on their forehead. All right. So amino acids, we're looking at this. Let's put them together. When we put them together, we call this bond is called a peptide bond. Peptide bond. Can I show you something? And then you're going to tell me what kind of process this is. You're going to tell me the chemical reaction that made them. So pay attention. If you don't, I'm going to take points away from you. How many bonds can carbon make? 4. We know, this is drawn a little differently, but we know that this carbon here is connected to an OH, right? I'm sorry, that I misdrew that. It's connected double bond O, single bond over here to this carbon, right? Right? Do you understand that I'm drawing this out here? This, and I'm drawing it out here. Does everybody understand that? All right. So follow along with me. It's a puzzle. So this is then connected for my 4th bond, OH. We got to connect it to this thing here. They got to be connected together. This has a full octet. It's set. Everything's set here. Everything's set here. So if we're going to connect them together using a covalent bond, something has to leave. Does that make sense? There's no seats here. We got to make some seats. So this carbon is going to be connected here. It can't do it with the oxygen. This carbon is going to be connected here. So we're going to get rid, now this is NH2. Let me redraw this NH2. NH. Okay, anybody guess what's going to happen?
Oh, the hydrogen will connect to the oxygen.
Yeah, beautiful. God, I love it when you pay attention. You are really brilliant. You really need to work on keeping that. Seriously. Do you know how few people get that right? Do you know how few people see it? Listen to me. I've been doing this a long time. Not a lot of people see it. You need to use that brilliance. She saw it. This hydrogen. And how did she see it? Was she born with this knowledge?
Mm-hmm.
Was there some emperor in the heavens that allowed a dragon to bring it down to her brain? I don't think so. She paid attention on a previous slide where we looked at condensation reactions, and she saw OH and CH, and she said what would come out? What would come out? Water. So we call that a condensation reaction. Dehydration synthesis. And you can go back in the video, if you're watching the video, you can see we literally just talked about this reaction. So now they're connected because these two leave. What can you do now with this carbon and this nitrogen? Yeah. You have your hand up. What was your hand? Oh, go.
So then it will be carbon and NH.
That's right. NH, just one H. So carbon and H. So what you get, the connection would look something like this. If I drew it up here, I'll draw it in blue. Now let me draw it in another color, red. So you get C double bond O connected to this one over here again, right? Connected to nitrogen, which has a hydrogen sticking out, which is connected to this carbon here. So now you have a chain. This is called a peptide bond. Do you have to recognize this? Do you have to draw this out? No. I could make you do it. It's not hard. But I'm trying to make things easy for you. I'm trying to keep things simple, straightforward. It's called the peptide bond. Why do I go through the trouble showing you this? For a couple reasons. One, you see dehydration synthesis. You see condensation. It's happening. Almost every time you make a polymer in your body, you're using the same system. You're pulling out water and you're making connections. It's just that simple. And the other reason is that you can understand why we call this thing a polypeptide.
Oh, that is wrong.
We call it a polypeptide because it has many what? Peptide bonds. A bunch of these together we call a protein. Someone close the door, please. Please do. You may have to open it again, depending if it's locked or not. So when we talk about a protein, all we're talking about is connecting them together. Now look at the different shapes. Do you see the different shapes here? That's because they all represent one of those 21 different R groups. There's 21 of these. These are going to become very important when we start talking about DNA and RNA and protein synthesis and mutations and all the rest of it. So if you can understand that each one of these R groups is different, that they're all connected in a chain of amino acids, if you can understand that why we call this a polypeptide, you're well on your way to being ready for your DNA section, your mutation, your protein synthesis section. So here are some of the amino acids. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. There's 20 amino acids here. There's actually a lot of amino acids There's actually a lot of amino acids. All right, there's more than your body uses. But these 20, of these 20, what do you think the star means?
It don't say it like that.
What do you think the diamond means?
It is essential.
Essential. Which means you have to eat them. You can't make them. You can make all the rest of them. But if you don't eat isoleucine, methionine, lysine, phenylalanine, tryptophan, threonine, and valine, if you don't eat those, and leucine, if you don't eat them with high quality protein, you're going to get sick or you can go blind. Right? Who went blind because they ate? Anybody? I heard people talking about it in the hallway. Who read about the kid that was eating Pringles and chips all day long? That's all he ate for a while. McDonald's French fries and Pringles. He ate some protein, but guess what he didn't? Guess what he wasn't eating?
Amino acids.
Not all those essential amino acids. It's absolutely, some amino acids, some fats are absolutely essential. If you don't have them, you die, you get sick and die. There's a story of an Eskimo tribe who saved an explorer, a European explorer. And the Inuit people have traditions. Do you know why they have those traditions? Guess why they have those traditions. To stay alive. And they saved him and they had him in the igloo and they were keeping him warm, him and his buddy. And they caught a seal. That's what they ate at the time, was seal. They cut the fat off the seal, the blubber. They cooked it up a little bit and they handed the blubber, just a big chunk of fat, to the person. And they ate their own fat. And they handed a piece of blubber. What do you think that person did?
They eat it.
They didn't eat it because it's not what he does. It's gross to him, right? I'm not going to eat a piece of fat. It's gross. They both died. Yeah. They can't. You need essential fats. They ate the meat from the seal. There's plenty of protein. But what's interesting about aquatic mammals like seals, they put all their fat in the outside of their body because it insulates them. So that's why we call it blubber, because it's a special way of storing fat. And all the meat they ate was extremely lean, very low in fat. And after a couple months of eating that way, could you stop? Not getting the essential fats that they needed, they died. So you need to eat a well-balanced meal, is my point. This is not an imagination. It's just a fact. You need a specific set of elements, a specific set of molecules. If you don't get these into your body, you will not survive. Or at least not in a healthy way.
How do vegetarians get that stuff?
It's all in vegetables. It's all in plants. Everything that we're talking about can be found in plants. You have to eat the right kinds of plants. You can't go around just eating broccoli all day long and thinking you're going to survive. You won't survive. Vegetarians have eaten, there's plenty of protein in lentils, what you call beans, or various things like various other types of vegetables that give you all the nutrients you need. But you have to do it. You have to eat that wide variety. You can't just eat what you like. Eating is not about what you like. It's about survival. All right. Amino acids. So you put them together in a bond. Let's take a look at some examples here. Neurotransmitters, neurotransmitters, central nervous system, glutamic acid. It's called GABA, right? Gamma amino butyric acid. Amino. A most common neurotransmitter of the brain. Others like glycine restrict to the spinal cord. So you actually use these things as every time you think. Right? Your brain uses these to feel. When you're feeling love, you're using these molecules. Yeah. This is a picture of a cell called a neuron, a brain cell. These are dendrites and axons. So this is actually a stained picture of spinal. This is actually from the spinal cord. All right, not the brain, the spinal cord. So here's red blood cells, normal red blood cells. Unfortunately, sickle cell, which we just talked about, the cell would look like that.
So half of the cell?
Sickled, like this, like death. The Grim Reaper has a sickle. Instead of being a circle, they look like this, and this causes a lot of problems. It gets stuck in the capillaries and causes blood clots, and it causes strokes and pain, and then just all kinds of problems happen.
I thought a sickle was more circular.
Yeah, it's not perfect, but it looks like a sickle. All right, so you can eat amino acids as a supplement. Do you see them at GNC? Is it time to go?
Yes.
All right.
We 1 minute late.
All right, so it's 1 minute left. Let me finish. Let me take attendance real quick. Let me see Carly before you leave. And what's next? 4th? And then we're done?
Yeah.
Please study this stuff. Study this stuff. You're going to get a quiz soon. Don't complain to me about not understanding if you're not asking questions. What is that? I don't need to sign it. I gave it to you.